Hi everyone. Good evening. How are you guys? So I hope you all have joined the class now. Okay, let's start. Let's not wait. So today's class will be on complete research cycle. Like we have been talking about, the research, how to start the research, and what are the, things we focus on the research. We focus, in the process of research. And in last class, we have talked about the ethics part, right? Ethics part, which is one of the important part of the clinical research. Today, we are taking a very specific topic which is like a little more advanced from that. First, we'll be talking about in this class, we will be talking about what is the research cycle, how the exactly research work from like how to start and how to end a research project. And then we'll be talking about like what is the types of what are the types of classes. I mean, what are the types of study designs? What are the types of study design? Okay, in today's class. And then in this class, there is one specific segment that is CDM process, clinical data management process. So in the entire research, see, yes, I got a question from someone. This is the first class of CDM. Yes, this is the first class of CDM only. Yes, this is the first class of CDM. So see, CDM is a part of clinical research. It's a part of clinical research cycle, right? So that's why I'm talking about. First, in this class today, we'll be talking about the research cycle, how to start research, how to end the research. And then a few study designs, what you must know. You all must know about those study designs. And then we'll come to the CDM overview, right? CDM overview means the process and the cycle of CDM. That also we will be talking about clinical data management. That is a specific part of the the research. Okay. So let me share the slides and let me start. So we'll take around 1 hour and 15 minutes class, right, guys? We'll take around 1 hour 15 minutes class. Let's start. Let me share the screen. Uh, okay. Please confirm if you can see the full screen of the, you know, please confirm, guys, if you can see the full screen. Okay, great. Okay, great. Thank you. Thank you, guys. So now let's start. So today, research cycle and clinical data management process. Okay. So definitely there are no conflict of interest. So as we always start, let's start with some questions to the audience. Yes, guys, tell me this question. So the question is, which of the following study design is the best suited for establishing a causal relationship between an intervention and an outcome? Intervention and outcome. How to establish the causal relationship between intervention and an outcome? Which study design would you be using to establish this relationship? Cross-sectional study, case control study, randomized control trial, cohort. Yes, guys. Answer C. Okay. D. Okay. Yes, guys. C. Everyone. C. Okay. Why C and why D? Justify. You have to justify, right? You have to tell me the justification with behind the right answer or the wrong answer. I mean, ultimately you have to give the justification for your answer. So why it is C? What do you think it will be C? Why? Vaishnavi Savari, do you take only one class every week? This was not my question. Anyways, no, we are taking some very, basic questions. I mean, sorry, basic classes first, and then so you understand the basics, and then we will start the advanced classes for everyone. Okay, so there is a process. There is a process of classes from basic to intermediate to advanced, right? So there are first some basic classes, what I am taking, right? So you must know these things also. These are the otherwise in the interviews, mostly you don't know the basic things. So this course, all the courses have very basic to very advanced, right? So yeah. Yes, guys, what is the answer of this? Why it is C or D? Tell me. Yes, guys. Yes, people. Why you are telling C and why you are telling D? Because this is a vaccine trial. Vaccine trial. Randomized control trial is a vaccine trial, Rahul Sharma. Is it? I mean, this is not a valid certification. Yes, because there is, see, if you look at the answers, guys, if you go back and if you check like what we learned in the last classes or what you learned in the clinical research subject, if you check only one word will differentiate the intervention, right? The intervention is only in the RCTs. See, cross-sectional studies, case control studies, and cohort. These all three are the study designs for observational, right? Observational studies. So that's why you cannot have the, I mean, this will not be possible for having an intervention in any observational study. So all others are observational studies. So right answer is, RCT, right? Right answer is RCT. Now, next question for you is in the research,
करके बैठ तू दोनों हेडफोन्स लगा ले हाँ लगा ले बंद
in the research, Vansika, you are keep yourself on mute, guys. Yes. So in the research cycle, which step comes after immediately after hypothesis generation? Data collection, literature review, statistical analysis, or study design and protocol development? Yes, guys. Who will answer this? Literature review. Okay. Anyone else? I think I have told you this in the last class also, like what is the research cycle? Little bit we have talked about it, right? Okay. Literature review. Yes, okay. B. Somebody is saying C also. Okay. Can someone write me the entire research cycle here? Can someone tell me the research cycle? How, from where it starts and where it ends? Any one of you? Can someone tell me what is the research cycle, guys? Fast. No time. Defining the problem? You can write in one message, not one by one. Write in one message. Yes. Research cycle steps. Anyone? Yes. Did you do any research projects? Last time you guys told that you have done many research projects also. So who are experienced here? Please tell me, how did you do the research? How did you do your research project? Tell me.
Sir,
Hypothesis.
first to identify the problem. After that, we'll go for literature search, and
Okay.
then aims and objectives, and then methodology, and after that, we'll go for data collection.
Okay. Okay. Where is hypothesis generation?
Sir, once we'll be analyzing the existing resources. Based upon the existing resources, we'll be coming to a hypothesis generation. From that hypothesis generation, we'll be doing the study design protocol, then data collection, literature review, and finally analysis.
Absolutely correct. Absolutely correct. So the step which comes immediately after hypothesis will be study design. Okay. Do you know that? I have told you in the previous classes also, guys. So how it start is define the question, define the problem. Okay. Remember this. I'm telling you again, define the problem. Then what? Then what you have to do? You can find out a question. Okay. You can frame a question based on that literature search, then objectives. Okay. And based on that, based on objectives, you will find out the hypothesis. Okay. Based on that, you will fix the hypothesis and then you will choose the study design and write the protocol, ethics approval, ethics approval, data collection, or data patient recruitment or data collection, then end of the story, end of the study, after ending of the study, data analysis, and then reporting or publication. Reporting or publication. This is the actual, cycle of the research. Understand? So hypothesis always come after your aim and objectives. And aim and objectives definitely cannot be defined without literature review. You know that. Without literature review, you cannot define the hypothesis. Understand? So the right answer here is study design and protocol development. Okay. Next question, guys. Next question. Which of the following best describes the null hypothesis? Null hypothesis. What is null hypothesis? Can someone tell me what is null hypothesis? Yes. Can someone tell me what is null hypothesis? My question is, what is null hypothesis? Rachna is telling D. Okay. Someone else who wants to answer this question and tell me what is null hypothesis? Yes, guys. What is null hypothesis? Answer the question. Null hypothesis. D. Okay. Chalo, fine. I agree that, okay, it could be D. There is no relationship between the variables under study. Okay. Can someone describe it with an example? With a case, I was supposed to say. Anyone? Pari, there is no relationship between the variables. That is written already. Null hypothesis is a question or the hypothesis that we want to prove it by doing our project. Mm, okay, we want to prove it. Okay. And if you want to prove that there is a relationship, then what is that called? In ideal research scenario, null hypothesis should always be rejected. Why? No, there is no such ideal research, ideal research scene. There is no ideal research scenarios because negative results are more impactful than the positive. Understand that. Negative results are more impactful than positive. Can someone justify this? If I'm saying, guys, see, I'm teaching you the practical part. I'm not taking you the theories. Okay. Tell me, if I'm saying that negative results are more impactful than positive, why I'm telling this? Yes. Anyone? Question after question. Negative results save others. Okay. And money, efforts and money. Okay. That's a great answer. Time and money. Okay. This is time and money. Time and money. Okay. That's good. Now let's come back. What is null hypothesis? What is hypothesis? Let's not talk about null. What do you mean by understand? What do you understand by, or what do you mean by the hypothesis? What is hypothesis? Assumptions. Okay, assumptions or expectations? Yes, assumptions or expectations, anything it could be. Yeah. Okay, theory. So, okay, see, suppose I'll give you scenarios. You have to answer those scenarios. There is a blood pressure. Like there is a group of people with hypertension, blood pressure, high blood pressure. A group of people with high blood pressure, and I want to check if their job stress contributes to the high blood pressure, if their job stress contributes to the high blood pressure. So what should be your hypothesis? And what is null hypothesis in this case? Just can you please explain? Can someone tell me what is null hypothesis in this case? And if we are doing this, how you are going to, which kind of hypothesis you are going to tell? Yes, guys, answer, come on. Let's be interactive. D is okay, not D. I'm asking the question. I have given you the case scenario. Discuss. Yes. Hypothesis is there is a causal relationship between hypertension and job. Okay. So what it should be? Positive hypothesis, negative hypothesis, null hypothesis, or what kind of hypothesis it should be? The job stress does not contribute to hypertension could be null hypothesis. Like if we are expecting that the job stress should not be associated with your hypertension, so it could be null hypothesis. Okay, null hypothesis will be there if there is no relationship between hypertension and job stress. Okay. Okay. Okay, fine. Okay, cool. So guys, if not null hypothesis, then what are the other types of hypothesis? Tell me. Yes, guys. Alternative hypothesis. What is alternative hypothesis? Guys, please turn off your mics. Yes, guys. One sec. What happens? Statistical hypothesis, the other possibility of research outcomes. What is that other possibility? Yes, guys, null hypothesis. Okay, see, listen. There are other hypotheses also. If there is null hypothesis, if there is no null hypothesis is just an assumption or expectation. Okay, null hypothesis is just an assumption or expectation what a researcher thinks that he is going to get this. Suppose I am doing a, I am doing a research in, suppose buccal cancer, and I have taken, 100 of the people, or 200 of the tobacco chewers. I took 200 of the tobacco chewers and I am following them up for 10 years. I am following them up for 10 years. So my hypothesis in the starting must be like, okay, this, the buccal cancer, the development of buccal cancer will be more in the tobacco chewers than the non-tobacco chewers, right? So when we are expecting that something is happening positively, right? That could be your, positive hypothesis. Okay, and if it is, null hypothesis is when there is no positive impact, there is no causal relationship, means that the buccal cancer, the number of buccal cancer is similar in the person in the group where they are chewing the tobacco and in compared to the non-tobacco chewers, right? So there we are proving that okay, there is no causal relationship between tobacco and the buccal cancer. Buccal cancer is happening similarly in the non-tobacco chewers also. So that is called null hypothesis, where there is no causal relationship or there is no relationship between two variables. Variables means there are two types of variable. One is dependent variable, one is independent variable, right? So if I talk about this only here, if I talk about job and blood pressure, raised blood pressure or hypertension, can someone tell me what is independent and what is dependent? What is independent variable, what is dependent variable? Yes, yes, guys, what is dependent variable, what is independent variable? Yes, someone. Independent variable like time? No, I'm not asking time. I'm asking in the job and hypertension, what is independent and what is dependent? Okay. So yes, job is independent variable here and hypertension is dependent. Okay, hypertension is dependent variable here and job is independent. What is dependent and what is independent is, see, independent could be many factors. Okay, it could be like, see, if the not job stress, then maybe, the alcohol consumption or high salt intake and the patient age, patient sedentary lifestyle, it could be multiple. So multiple independent factors, affect the dependent factor is always one, right? When we are doing in the research, when we are taking care of the research, there is only one dependent factor. There is only one dependent factor. Okay, there is only one dependent factor and others are all independent factors. So independent factor can affect each other also. Independent factors can affect each other also, like, diabetes and the sedentary lifestyle, or diabetes and alcohol, okay, and hypertension. So hypertension, if you're talking about diabetes, considering as dependent factor, then hypertension and alcohol both can, relate, like the intake of alcohol can cause hypertension also, and hypertension and the alcohol consumption both can cause diabetes also. So there is a causal relationship between even in the independent variables. So when we check, that is a statistical part, I'm not going to that, but when we check the regression analysis or multifactorial analysis, we check all these independent factors one by one with the dependent factor, and then we check all them together, and then we check the effect of each independent factor on each other, right? So that's how we do the research to find out the very precise results to that to prove or unprove the null hypothesis, like in the clinical trials where we just work on the very low connections, like maybe the p-value 0.0012 to 0.049, right? So that kind of things we talk about when we do the statistics, right? So in that cases we go for the group analysis, subgroup analysis, and in subgroups also we go for the variable analysis, multifactorial analysis. So like we just try to find out the the lower level of connections. Maybe at the bigger side, there is no connection. Maybe at the bigger side there is no connection, but sometimes there are the relationships between two variables at a very strand level, okay, very minute level, right? So we have to get into the different, like suppose I'll give an example here. Maybe the gender directly does not affect the heart attack. Gender normally, okay, gender normally is not directly associated with heart attacks, myocardial infarction. Focus on this. These are the very real time examples what I'm giving. Okay, so gender may not be directly associated with heart attacks. Gender means when male, female, and coming. But when we stratify gender, when we stratify gender into male and female, then you will find out that females are more associated with the myocardial infarctions than the males, right? So there are a lot of things where you see, suppose maybe age, if you stratify more, you will find out the elder people are more associated with heart failure, younger people are more associated with the maybe the congenital heart problems, or maybe the valvular heart problems. So that's how we stratify, and that's how we try to get into the deeper statistics, and then we try to pull the hypothesis there as well, right? So the null hypothesis basically is when there is no connection, but when you find out the connections, you go to the another hypothesis, alternative hypothesis, and you can to check that you can stratify the data also, you can stratify or subgroup the data also. Okay, so there are multiple things what you do usually, but if it is only about the hypothesis means the connection, hypothesis means the assumptions or the expectations, what we feel, what we expect to happen after our project or after our research, that is called hypothesis, right? So hypothesis can be positive, hypothesis can be the null hypothesis where there is no connection. Okay, if there is connection, that the connection could be negative or positive, both types, right? Suppose in subgroup, suppose I'll give an example here where you will see the this is not null hypothesis, that is not called negative hypothesis or positive hypothesis, but the connection relationships, how we defined the relationship is called as relative risk. Okay, sorry, relative risk. Okay, that is called as relative risk. This is another term what you have to understand, relative risk, but that is another part of the connection or relationship, right? So suppose they see we call it 1. The normal value of relative risk should be 1. Guys, focus here is very important. Relative risk normal value should be 1. Normal value 1 means, see, there is no connection between, there is no effect of treatment A on B, right? I mean, treatment or A on disease B or whatever, there is no effect. Or the group A or variable A is not affecting variable B directly. Okay, there is no effect of variable A on B or variable B on A. There is no effect. That is called as 1. That is called as 1. Relative risk 1, like, see, like 50-50, like 50-50 chances, like there is no specific chances higher on any side. So this is called 1. Relative risk 1 means there is no chances of relationship between two variables, right? But if it goes less than 1, if it goes less than 1 means, suppose less than 1. So how it will go less than 1, guys? How it will go less than 1? Less than 1 will go only in case like this, 40-60, right? Or suppose very minute, 49-50, right? So then in that case it will go lesser, right? So the thing is here, it will be like the effect of B, effect of B is higher on A, right? Effect of B is higher on A. Suppose A variable and B variable, right? A variable and B variable. So this is called negative impact. Negative impact. Negative impact means, suppose I'm telling you here, see, now I'll give you an example so you can understand. Salt intake, right? Salt intake and hypertension, right? Hypertension. If the relative risk is 1, that means salt intake has no effect of on hypertension. Salt intake has no effect on hypertension. If the relative risk is less than 1, less than 1, suppose less than 1, like suppose 0.8, 0.8, that means salt intake is protecting from the hypertension, because see, salt intake is better for the hypertension. Like salt intake is better for the person to reduce the hypertension. If the relative risk is less, if the relative risk is less, that means that there is negative impact. Negative impact means the salt intake is not causing the hypertension, but salt intake is protecting from the hypertension. Okay. And if it is more than 1, like suppose 2, that means the salt intake is increasing the chances of chances of hypertension by 2x, 2 times. If it is 3, 3 times. If it is 4, 4 times, right? So it is a positive impact. It is a positive relative risk. It is a positive impact or positive relationship. Understand? That's how we divide the relative risk, and based on that only we usually come to the hypothesis testing. Hypothesis testing is actually known by p values, right? Like 0.05 is the normal value, and less than 0.05 is the positive impact. Like, yes, salt intake is causing hypertension. If it is more than 0.05, that means okay, it's negative impact, or there is no impact, right? Like that we decide, right? So p value is one of the major part of deciding the null hypothesis. But there is no only p value. People just talk about the p value, but p value is not only the one element which is helpful in testing the null hypothesis, but there is also relative risk, and relative risk tell us about the positive and negative impacts. Understand? Understand, guys? It is clear for you or not? See, if we go, we can take 2-3 lectures on null hypothesis. But I was just describing you in a very brief thing, like you can understand what is null hypothesis, what is relative risk a little bit. So it causes, it creates okay, fine, right? So the correct answer here is no relationship, right? There is no relationship between the variables under the study. Perfect. Okay. What is the most appropriate study design to explore the prevalence of hypertension in a population at a single point in time? Yes, guys, in a single point in time. C, C, B, case series, okay, cross sectional. Hmm. Hmm. It is C. It is cross-sectional study. Cross-sectional studies are only studies which can be done on a single point of time. Single point of time studies are known as cross-sectional studies. Okay. Cross-sectional studies. Cohort are longitudinal studies. Cohort are longitudinal studies where we have to wait for years to get the result or to prove the hypothesis. Okay, and randomized control trials are also longitudinal interventional studies. Okay, so that the correct answer is cross-sectional studies. Cross-sectional studies. Okay, then yeah, cross-sectional studies. Next question for all of you is, I think last maybe, which phase of clinical trials primarily assesses long-term safety and rare adverse events in a large population? Phase 1, 2, 3, or 4?
Phase
Yes.
4.
Yes. Phase 4. Okay,
Phase
someone was
4.
telling phase 3 also, but it is phase 4. It is phase 4 because phase 4 is also known as what? It is known as what also?
Post-marketing surveillance.
Post-marketing surveillance. Fantastic. Post-marketing surveillance. So post-marketing surveillance always covers the most of the population. Okay, post-marketing surveillance covers most of the population, and that helps in checking out the real-time adverse events in the public. Real-time events, because see, when the clinical trial is happening, that is happening in the restricted environments, in the restricted population, which have been selected to the selected to the clinical trial. But when after the marketing, post marketing surveillance, we call it, suppose, so when the post marketing surveillance is done, that means when the drug is already been prescribed to the patient and we don't know what type of patient, right? Based on the clinical judgments of the doctor, clinical judgments of the clinician, we give the drugs, right? And based on that only, those patients are taking these drugs, and they are assessed for any kind of long-term side effects. Like, can someone give me an example of long-term side effect of any drug? Long-term means at least after 3-4 years of taking the drug. Yes, guys. Hepatotoxicity of, renal failure of. No, renal toxicity is okay. But tell me the drug name also. I'm asking the example of drug and their long-term effect. Yes, guys. Renal toxicity is okay. Hepatotoxicity of paracetamol. Okay, I mean, it's a debatable topic, very nice topic. Can someone tell me diclofenac or whatever? Diclofenac or whatever. How much of diclofenac for how many years can cause renal failure? Yes, everyone. In which cases the diclofenac or the paracetamol causes renal failure? Yes, guys. Diabetic medicines causes neuropathy. Is it? Okay. Or diabetes causes neuropathy. Yes, guys, diabetes cause neuropathy, or I mean, diabetes causes neuropathy, or diabetic drugs cause neuropathy. Statins cause muscle break. See, these you are just read somewhere, or you read in the classes. Tell me some good examples. Good examples means with the timelines, proper timelines. Why? Osteoporosis due to steroids. If I'm taking, prednisolone for 5 days right now, and if I take prednisolone again for five days, after 6 months, and again for 5 days after 6 months, does it cause osteoporosis to me? Does it cause osteoporosis to me?
Each phase of clinical trial has long-term safety and rare adverse effect.
So long-term effect means what long-term effect? How much long-term effect it needs? What is the quantity should be there? What type of long-term effects? Guys, come on. This is simple. Give me some example of that long-term effect. What is like, how it is? Long-term steroids causing Cushing syndrome. Cushing syndrome. Okay. How much long-term of steroids cause Cushing syndrome? See, there are always, this is a conclusion what you are telling. This is a statement what you guys are telling here. You have no scientific, proof with you right now. If I ask you what long term, which study says, these are just statements. You should have something in your hand like, okay, sir, if I'm talking about steroids, see, steroids means what? Steroids, if I'm taking prednisolone, I'm giving you the question now. I'm taking prednisolone every 6 months for 5 days for next 10 years. Would it cause kidney injury to me? Answer. Would it cause kidney injury to me? If I'm taking 5 days prednisolone every 6 months for 10 years, would it cause kidney disease to me? Yes or no?
Yes.
Okay. Why yes? And do you really think is it a long-term use? If I'm taking,
Prednisolone is a steroid, sir, and
yes,
no, it is not a long-term use.
if it is not a long-term use, how it will cause the kidney problem? Yes, guys. See, people, this our girls take this tablet. What's that? Mefenamic acid, not mefenamic acid, right? What is that? Mefenamic acid, right? For the menstrual cycle pain. That is mefenamic acid, right? So, see, people tell that mefenamic acid, this meftol tablet, causes the kidney injury. Causes the kidney injury in the ladies or in the people who are taking it for the long term, right? So suddenly, all the girls, all the ladies has stopped taking meftol for their periods pain, right? But that's not correct. That's not correct because they are not using it for the long term. They are using it on the intervals. Like they are maybe taking 2 tablets or 3 tablets every month maximum. See, where they got the scientific evidence from is, see, the people got the scientific evidence of meftol or mefenamic acid induced kidney problem is in cancer patients. Do you know that? When the cancer patients after chemotherapy, they have the continuous pain for months, right? And they are always in pain for weeks and months. So that time they take a very high intense doses of diclofenac or paracetamol, like they take grams per day. Like this tablet 500 mg you take in a day once only, they take 4 times or 5 times in a day for months. So in certain cases that causes the kidney injury. Understand? In that cases, it causes the kidney injury. Otherwise, it does not cause the kidney injury. And see, that's because the someone has given the statement that long-term use of mefenamic acid is causing the kidney injury. So all the girls, all the ladies has stopped taking meftol. Now everyone is like, no, ma'am, we don't take meftol now. We can bear the pain or we can take another options, but not meftol. No, first you don't understand the concept behind it. First, you don't understand the concept behind medicine and how to take the medicine, right? So taking 2 or 3 days in a month for the specific period does not cause kidney problem, because once you take these 3 days or 3 tablets, first of all, that is not causing any harm to the body. Okay, that is not causing any harm to the body. And these 3 days will just, wash out in 2 to 3 next days. 2 to 3 days, it will completely wash out from your body. Understand? So there is no pile up of the concentration. There is no high concentration of mefenamic acid in the body, right? So why we are discussing this is because when we do clinical trials, we do it for a short term time, right? We do it for, a month or 6 months or 5 months. But there are some drugs which cause the injuries or harm to the body when they are, being taken for years, long term effect for years, like 4 5 years, 10 years, or 15 years, then only they cause the problems, right? For an example, metformin, if it is taken chronically, like suppose someone is diabetic in 2015 and he took metformin 500 mg for 3 years, then metformin it was not controlled, then metformin 1000 mg for again 5 years, and then they are on metformin 1500 mg for last 3 years. So the metformin can start causing now vitamin B12 deficiency, right? So that is called long term. So high dose, more than 1 gram of metformin, there should be always evidence and numbers. More than 1 gram of metformin every day for at least 4 years, then there are the 60% chances to get vitamin B12 deficiency. That is post-marketing surveillance. That is post-marketing long-term registry studies or long-term effects. That's how you should define, that's how you should know the context. Metformin, otherwise the people who are taking metformin, they will stop taking metformin at all. They will not take metformins. Oh, metformin causes the vitamin B12 the deficiency, it can cause me anemia. No, because the context says the specific numbers, context says the specific percentage, right? And then based on that, you should decide if it is being taken or not, right? Not like I'm taking, maybe if I'm taking one tablet of diclofenac or one tablet of paracetamol every week for my headache. So I'm just taking 48 tablets in the year. That's all. And that is also on the frequent, not that is also very not frequent. Those are occasionally, once in a week. That is not causing the kidney injury to me. Right. So that's how you should know what is the impact, what is the timeline, what is the duration, when they did the study, in what scenarios they did the study, what was their population, what was their timelines, right? So then only you should believe on the facts, not on the statements. And that's why we need larger populations, and we do in the most marketing surveillance that is called as phase 4 to know the long-term effect on the drugs, because the actual concentration is piled up in the body after years, and then the continuous concentration can cause the problem. Antibiotics, yes, some antibiotics are being resistant because antibiotics are quite what we say quite sensitive drugs, and they can cause even the resistance in 10 days 15 days, and 10 days 15 days not even immediately, but after if you are taking frequently, like every 3 months, every 2 months, again antibiotics are not even, if you are taking antibiotics even every year, they will not be resistant. That they will not be resistant. When they get resistant is like, suppose you get you are sneezing, you go to the doctor, take 5 days amoxicillin. If you go to the after 10 days you got throat pain, go to the doctor, 5 days amoxicillin. If you again get the stomach pain, oh you have stomach infection, again 5 days amoxicillin. So if you are taking very frequently, like 10 days or 15 days course in a month, then only it get resistance. Okay, even the antibiotics get resistance only when you are taking very frequently, and the full dose of antibiotic, full course of antibiotic, then only it causes the resistance. Understand? So you have to understand. That's why there are the 4 phase. I mean, this phase 4 clinical trials or post marketing surveillance, which actually causes and which actually tells us about the long term effects of the drugs. Okay, guys. So phase 4 is the correct answer. Yeah, so this one is the about CDM. What distinguishes electronic data capture systems like Medidata Rave and RedCap in CDM, clinical data management? What distinguishes electronic data capture systems like Medidata Rave and RedCap in CDM? So the answers are here. You have to choose the right answer. They eliminate the need of case report form. They automatically generate case study, clinical study reports. Okay. They allow real-time remote data entry by investigators. They are used primarily for the safety reporting of FDA. So why? Yes, guys. So what could be the right answer? Like why we choose electronic data capture systems? Yes. B. Okay. Automatically generate the case report forms. Okay. Anyone else who wants to answer this? How many of you have known to the electronic data capture systems, EDCs? How many of you have worked with EDCs before, guys? No, answer will be okay, fine. We'll discuss this question first. How many of you have worked on EDCs? Anyone who has worked on EDC before? Okay. What was your project and which project did you do? Tell me, guys.
I have worked on Medidata Rave.
Under the Medita Rave. Okay. For
Yes, sir.
what purpose?
For a study purpose, like for the project, the organization.
Okay, it was for project organization. Some NGOs are using the red cap and all. Yeah. Okay. Okay. So guys, the correct answer is definitely C. And why the correct answer is C? Because see, See, the main problem in the clinical research comes when you have multiple centers. Okay, clinical research, multi-centric is a very tough job, very tough task, very expensive task. So suppose I'm doing the clinical research, which is expanded to 10 centers in India, not outside. I'm just considering India. Like one is in, Manipal, then second is in Delhi, Ahmedabad, and Chandigarh, and Lucknow, Udaipur, and various centers, Chennai and Bangalore, right? So they are you know recruiting the patients for me. I'm recruiting the patients at the main center. Now how would you connect with them? How would you get the I want the final data sheet at the end for the analysis, right? How would I get that seat? How would I get the references? How would I get the numberings for the patient? I don't know, like which one is what number. Like suppose today 10 patients got recruited from different sites. What patient was at which number? Okay, we don't know. We don't know the live tracking. So the best part here is real time remote data entry by investigator is being recorded in the EDCs. So EDC has multiple logins. Every center will have the login. So suppose one center at 10 a.m. is recorded at Chennai center and 10:15 one in Chandigarh. Everything will be in one database with those requirements, and it is a real time access. It is a real time access to everyone. I mean, they can fill the forms, they can update the data, they can enter the patient to the EDC's electronic data captures. And based on that, it is very helpful in combining the data, defining the data, and monitoring the data. Monitoring the data means monitoring the number and how many patients have been recruited from this center, what are those patients, and if there any discrepancy in any patient. So the team which is sitting at the center can easily monitor on time, right? Those teams. So this is a process. You must understand when you are signed up for this 3-in-1 or 7-in-1 of diplomas or clinical research. You must know the process and what exactly happened at the center. Right now maybe you are just you have come out of the studentship, you have been a student, and you don't know how exactly it work for the industry. For the industry, there is a huge system. There is a huge system. I have been I have worked as a principal investigator for a multinational clinical trial. I've been working for 7 clinical trials. If you might have heard of the names also. Paradigm clinical trial, and this another clinical trials for the heart failure drugs, this sacubitril valsartan, right? So paradigm and paragon clinical trials, I have been working as investigator in those clinical trials. So there are the different systems, RMS, multiple millions of the dollars have been spent on these databases, electronic databases, to capture the data from. There were 432 sites, 432 sites from 100 plus countries, and that's how they manage it. So EDCs are something which collects the data, and they allow the real time and remote data entry by the investigators. Remote data entry is needed because they have to put, if I'm recruiting the patient today, there are the rules that within 12 hours you have to update the recruitment, right? You have to update within 12 hours or 24 hours. There are the different rules for the different clinical trials. So you have to follow those rules, right? So EDC is known. See, now you will tell where we are comparing. EDC systems are top, they are distinguished from others, but where we are comparing, see, we are comparing with the paper format. See, usually at every center, when you do the clinical research, you make the Google form, or you either make the paper format, right? So you take the data, you collect the content, you collect the data, patient related data in a paper format, and after the paper format, then you enter into that, and then you will make a Excel sheet, or then only you will send the Excel sheet or something. Or I mean, lot of problems. You have no real time data capturing, you have no like live monitoring, you have no discrepancy management, you have no CDM management in that. So that's why these trials have become, I mean, after the lot of evaluations, sorry, lot of evolution, we have come to that, we have come to a final thing, like, okay, we have the EDCs, and now different companies use different, like Medidata Rave is there, Redcap is there, and other data, RMS is there. Previously this IQVIA has, previously it was RMS, okay, and that RMS has changed to something else, and then IQVIA now. So we used to do in our RMS. That is that is also for the research management system. Okay, there is also, there are DMSs, data management system. So multiple things are there which are there in the market. But right now this rave and CDM के लिए, I mean red cap are the one of the world famous things what everyone uses. So the answer here is this allows the real time remote data entry by investigator. Okay, now let's come to the topics where we will discuss most of the things we have discussed already, but let's still focus on these things. So research cycle. So you can see here. Research cycle starts with the defining the issue. Okay, research cycle starts with defining the issue. Okay, someone's mic is on. Can you please look into your mics? Always I want to tell you guys, huh? So this is first is find a relevant emerging issue that links to both the organization and the individual needs. So first of all, define the problem. That means define the problem, right? You are defining the problem. Then it goes to building a question. As I was telling, after problem, you will be building a question, and based on the question, you will be doing a literature review. Okay. Then you will be doing a literature review, and after literature review, you will be refining that question to aim and objective. To aim and objectives. Understand? You will create the aim and objectives. This is literature review, right? And once you have the aim and objectives, you will definitely have the hypothesis. They have not written hypothesis, but the question of a hypothesis, you will have the hypothesis. After hypothesis, you will select the methodology. You will select the methodology from multiple types of methodologies, and then you will go to collect the data. Based on collection of data, you will be analyzing the findings. And then you will be reporting the findings, like share the new understandings, like reporting or making the thesis or making the publications, publish it. Okay, and then adapt or affirm. So adapt or affirm your own practice in light of what you have been learned from that, right? So you make the report. Again, that is also the part of reporting, right? Report it and get the feedback done. Okay, so this is the actual research cycle, guys. That's how you start the research, any type of research, whether it is industrial research, the process. This is just a framework. This is just the outline. This is just the outline or a framework. This is not something which is actually happens in the see. Suppose it's an institutional research project. You will come up with a question directly. You can come up with a question directly with your friend's help, with your guide's help. Your guide will give you the topic, and then you will write the methodology direct. You will not go to the literature review. Maybe directly you will go to the methodology, you will go to the protocol writing, and you will recruit the patients, and you will submit the thesis. There are only 3-4 steps you will follow. But when it is industrial, because you are in the industrial trainings, right? So when you go to the industry, each step matter. Okay, these step matters, and in these steps only there are another multiple steps, right? Multiple steps. So if I talk about just building a question, there are many meetings happen with the collaborators. First, they will define the team, they will define the director, they will come up with the problem, they will allot the some starting budget, right? Okay, they will establish the facility. Okay, this facility will work on this. They will establish an initial team. They will come up with the proposals, and then they will create a team, and then they will come to the problem, and then they will come up with a question. And once you have the question, then the another team, the review of literature will be done. Review of literature will be done by the entire medical writing team or medical affairs team, and they will be creating the PPTs. They will be creating some other team will be creating the mesh terms, and then they will go to the literature search databases. They will do. There is a huge timeline involved. There is a huge money involved. There are many people involved. The time, and then once they have the relevant question, once they have done the review, what they will do is they will refine the question, they will set the hypothesis, then finding the aims and objectives, and aims and objectives, and then they will create they'll create the protocol. Okay, they will create the protocol. Not only this, in the protocol creation also, they'll be like, someone will be talking on the risk, someone will be talking, someone will be working on the what we say, compensation, someone will be working on the other parts of protocol, right? They will be collaborating, someone will be developing the EDC, someone they will have the clinical data management team which will be developing the CDCs. They will be, by the time they will be selecting the sites, going to the sites, managing the sites, they will be hiring the CRAs. A lot of work is there. Lot of work is there. The institutional task, these tasks are not very simple. Conducting an industrial clinical trial is not at all simple. It's a Herculean task and long-term task with a high, very, very high budgets. You know, I'm telling you about the Paragon and Paradigm. One clinical trial, phase 2 and phase 3 clinical trials of Paradigm and Paragon. One was, like the expenditures of one clinical trial in 412 sites was around $300 million. You know, $300 million on one drug clinical trial worldwide. They used to pay 36,000 for one patient. 36,000 for one patient to the investigator and investigation team, and they used to pay the compensations, they used to pay the, like travel charges, food, to the patient, and their free tests, everything. So like when it comes to the clinical trials, that is not just a student project or institutional project. That becomes a huge task. That becomes a huge task with another hundreds of the things included, right? Hundreds of the another things included, right? So that is, very important. One sec. Okay, so that's how the industry clinical trials have been conducted, right? So these all things, research cycle is an outline. You should understand what exactly and how exactly it works, right? But if you're reading that, that will be done definitely in the clinical research. You will be getting all the things in, the courses. Okay. So see here, the same thing, defining the problem, literature review, formulate the question, develop the research conceptual, research methodology, data collection, analysis, finding discussions, research publication. Okay. Now, types of study designs. Can someone tell me what are the types of study design, guys? Yes. Can someone tell me what are the types of study design do you know? Yes, everyone. Types of study design, guys. Let's talk about study designs. What are the study designs you know? Yes. Experimental observation. Okay. Anyone else? Last time, what are the types of study designs or study types I told you guys? Any idea? Case report, cohort. Okay. Cross-sectional, RCTs. See, as I told you, many types of studies are there, first of all. If you based on the timeline, if you look at the, timelines, one is forward timeline, one is backward timeline, retrospective, prospective studies, retrospective and prospective studies. Then, based on the intervention, there are two types. One is without intervention, those are called as observational. One is interventional studies. One is interventional studies, another is observational studies. In observational studies, there are multiple types like case report, case series, cross-sectional, cohort studies, and this what we say, case control is there, cohort is there, cross-sectional is there, and then case report, case series, and interventional. There are randomized control trials, community trials, and non-randomized control trials. So multiple things are there, but there is something like this also, what you are seeing in your screen, right? Experimental designs, correlational designs, diagnostic designs, cross-sectional designs, action designs, longitudinal studies. So these are different things for different terms also, what where we use, right? Quantitative, qualitative, and mixed methods. So like descriptive, experimental is interventional, right? Experimental means the interventional studies. So it examining the cause and effect relationship was we were talking about. So causal relationship is assessed by the experimental studies. Then correlational studies are there. These are identifying the relationship between variables without manipulation. So they just check only correlation, not the relationship. Okay. They check the correlation, not the relationship here. Diagnostic means these are defined only for the diagnostic purposes, like diagnosis of any disease, or diagnostic biomarkers, or diagnostic tools or diagnostic procedures. So the studies which are designed only for diagnosis are called as diagnostic designs, and those designs are quite different from the normal designs, because maybe in the normal designs we use the patients patient's data, retrospective and all. But for the diagnostic, we have only, certain designs like RCTs and all, right? So diagnostic designs are different. Okay, like the companies like Rose and these companies which creates the, diagnostic kits and diagnostic tools, they use those diagnostic designs. Then cross-sectional, of course, point of time, at one point, right? At a point of time, then case studies or case studies. Someone's mic is on, guys. Okay, and then descriptive studies are there. Describe the characteristics of a population, like prevalence and incidence understanding. So those come under the descriptive studies. Okay, those come under the descriptive studies. Okay, and then longitudinal means for the long term. These longitudinal are long term. Exploratory means they are like just to, before the clinical trial or we do the pilot studies. Exploratory are the pilot studies which are done to get the initial knowledge or initial insight on any topic. Then action to implement the solutions, like to implement the policies, to implement the treatment, or to implement something in the companies or in hospital or in the population. So those implementation studies are called as action studies. Then the mixed methods, where we mix two methods, quantitative and qualitative, those are called as mixed methods. So these are the just terms, but you must understand this is the actual thing what we were talking about last time, and I have described it. So there are two major types of studies. One is one is analytical study, one is descriptive. Okay, one is descriptive, one is analytical. So descriptive means only case reports, case series, and descriptive surveys, where we just have to describe the population, like in the the characters of the population, where we just have to characterize the population. We have to describe. There is no statistics applied. So in descriptive studies there is no statistics. Remember this, the difference here is no statistics applied. Okay, and in the descriptive, again analytical, if you go, there are observational. So guys, one more thing, these all are also observational studies. Okay, these all are also observational studies. You can connect like this also, this then this type. Okay, these are also observational studies. These all are observational studies, this and these all are observational. Descriptive are also observational studies, right? So in observational there are cross-sectional studies. Cross-sectional studies means like at one point of time. Case control means where we compare between cases and controls and we come to a conclusion. So case control studies are usually backward. Okay, backward studies. Like there are two things. I'll let you I'll explain it here so you can understand. See, last time also I told you there are two terms what you have to understand majorly. One is exposure, one is outcome, right? One is exposure, one is outcome. So both are the terms we will be using in the entire clinical research, and the entire clinical research is depending on the exposure and outcome. So exposure means any causative agent, any causative factor, or any causative factor which can create any problem, which can cause any problem. That is like, that is a that is called as exposure. Suppose for an example, suppose for an example, this what we say, smoking causes, what is that? Smoking causes the lung cancer, or buccal cavity cancer is caused by the what we say, what we're talking about, tobacco chewing, right? So these, in this there are exposures and outcome. Outcome is a disease and exposure is any element, like tobacco and buccal cancer, smoking lung cancer, right? Stress, heart attack, right? So stress is exposure, outcome is heart attack, right? So that are the types of things what you have to learn, right? So based on that, there are two types of research studies. There are two types of research studies based on that. Now I'll describe that. See, first, you went to a society or you went to a village or anywhere where you met 1000 of the people. Guys, carefully understand that you found 10,000 of people who are smokers, and you don't know that I mean, you want to check like how many of smokers will develop cancer, will develop lung cancer in 5 years. You are doing a study in smokers and you are finding the outcome, like in 5 years how many will be doing the cancer. And if you want to establish that, you will be taking non-smokers also, you will be taking non-smokers also. I mean, it's not necessary in that case. You'll be just checking the prevalence and incidences of lung cancer in smokers next 5 years. So next 5 years, when I'm talking next 5 years, that is the forward direction studies. Forward direction study means we have the exposure, we are looking for outcome. Focus on that. We have the exposure, we are looking for outcome, right? This is called as cohort study. This happen, forward studies are cohort studies. Okay, forward studies are cohort studies where like we are having the smokers, but we are looking for cancer, right? For 5 years, 10 years, long-term longitudinal studies. Those are cohort studies. But when we suppose there are 100 people, there are 100 people with cancer, but we don't know how the cancer has happened, how the cancer has happened. So what we will be doing is we will having these 100 cancers, we will having these 100 non-cancer. Now we will, these are our case, controls, these are our cases. Now we will go back, we will go back means we will go to the exposure. Now right now we have outcome with us and we are going to exposure. We are going backward actually. If I mean the non to just to show you, we are going backward, we are going from outcome, we are going from outcome to exposure, right? We are going from outcome to exposure. So these type of backward studies, when we do, we need a case and the control to compare. Like these 100 cases, what has been wrong? So these all 100 got the lung cancer. What has been wrong, right? And in the healthy patients, what has been, be the corrected part or element, like they did not get the cancer, right? So you will find out that they all were the smokers, almost all were the smokers. You will find out the ratio, you will find out the negative relative risk or positive relative risk. So here we are going back. So always remember, cohort are forward and case control are backward design studies. Case controls are always backward design studies. Okay, backward design or backward direction, back direction studies. Okay, back direction studies. Case control are back direction, cohort are front direction, cross sectional are point in time, right? Point in time, and these all are observational and these are interventional. Interventional means all the type of clinical trial, randomized control trial, non-randomized control clinical trial, single arm clinical trial, double arm clinical trial. So I'll show you here. See, these are the analytical studies. That's especially I brought for all of you. Okay. So analytical study means where statistics has been applied. So in the observational study, in these three also statistics applies. Statistics applies, but not the inferential statistics. There is something called inferential statistics, inferential, and there is something called descriptive, right? Descriptive statistics and inferential statistics. So in the observational studies, these three types, only descriptive study applies here, like non-parametric test, parametric tests. And non-parametric test means actually not non-parametric, but parametric test, like the pair t-test or independent t-test, or maybe the ANOVA, right? Those tests has been applied here, prevalence, incidences, chi-square test. So this is called descriptive study. But when we go to inferential, here you will find out the multifactorial, or multilinear regressions, linear regressions, or all these things you will find out here in the experimental studies, right? That is different. But in experimental studies, another major characteristic is intervention. Right, you will have intervention here, you will have no intervention this side. So intervention means experimental, one or more factors are altering the major effect, right? Those are interventions. So based on that, like uncontrolled clinical trials where there is no control, like where there are no specific arms, like which will take what treatment or cases. Controlled trials means there is a treatment, there are cases. And in the control trial also there are two types, randomized and non-randomized. So randomization and non-randomization are the techniques of hiring the patient or recruiting the patient. If you are taking the patient randomizingly, randomly you are taking anyone into the study. Nowadays randomization is computer based, randomization is application based, right? So if you are taking randomized patients into the study, that is called randomized clinical trials. If you are taking non-randomized, that is like if you are taking without randomization, that is called non-randomized clinical trials into the cases, cases or control. So these are all intervention clinical trial, right? So that's how you define the case studies, and that's how you understand the case, the research designs. Okay, so these are the research design types normally. Okay, now CDM, if I just talk about the CDM, there are the CDM is the essential task. Yeah, somebody's having question. Anyone has the question right now? Sir, in descriptive statistics, only central tendency or dispersion. See, dispersion and central tendency, along with that, there'll be some kind of normalcy test, or the parametric test also applies in the descriptive task. Okay, usually in descriptive, some kind of parametric test also we can do, like the pair t test, or independent t test, or ANOVA or chi-square t test. Those are also can come under the descriptive statistics. But usually, yes, prevalence incidence, descriptive studies are talking about central tendency and dispersion. But in some, studies like observational studies, like, cohort studies, case control, you need these non-parametric, I'm sorry, these parametric tests. Okay, so CDM, clinical data management, is part, guys. CDM comes when you start collecting data. So CDM starts, see, not only the collecting data, the part of clinical data management. See, the people who are learning clinical data management here or anywhere who wants to learn CDM, their part starts from the very beginning. Their part starts from the very beginning, from where they will start thinking, or they will start creating the CRF. CRF is case report form, right? So they will, I mean, the CDM means they will be working on the data completely. They will be working on the data. So data is not just that you will get the Excel sheet, you will clean it, file it, and analysis. No. See, CDM means when there is a, research comes into the existence, the research topic, from that time only the CDM team will be involved in, like, what kind of data will be collected, right? What kind of data will be collected, and CDM team works in the data form creation, case report form creations, and then making them into the, digital part, like EDC, electronic data capture forms. And then, like, they will be giving the access to the sites, they will be collecting the data from the sites, they will be refining, they will be analyzing, they will be, they will be having the security of the data, they will be, maintaining. But so there are multiple tasks. CDM team involves from the first step. CDM team involves from the, designing of the study, designing of the entire clinical research project, to the end, till publication, and even to the retrieval, or to the archival. CDM team is even, working after the clinical trials in archiving the data, and storing the data, right? So CDM is not just just someone who will work on the data and just like that, but CDM works from the first phase to the last phase. Okay, so CDM is essential part definitely of the clinical research. It involves in the collection, cleaning, management of the data from clinical trials to ensure the quality, reliability, and compliance with the regulatory standards. CDM ensures the clinical data is collected, processed, and maintained to facilitate the accurate analysis and reporting. So main characteristics of the CDM is accuracy. Definitely they have to maintain the accuracy. And see, that's why the real time, data collection and real time surveillance is important, because they are collecting the data from different sites. So they have to make the unique type of data sets, they have to make the unique EDC, so everyone fills that format only, everyone fill the data in that format only. And the central team, which is sitting in some central, facilities, they get the unique and, similar data from every site. They get the similar data from every site. That's why we need the CDM team. So accuracy, consistency means, see, uniformity in the data formats and structures across the trials. Across the trials means even the sites they're talking about. Integrity, safeguarding the data from unauthorized access or alteration, so nobody can alter that. They have the data locking systems, they have these things. You will see in the CDM classes, you will learn about those data locking. Then they follow the GCP, good clinical practices, to adhere to the data and for the security of the data, and other guidelines like from ICH and FDA, right? So this is the clinical data management process, whole data management process, like where they exactly work. Guys, this is very important, what you should understand and how you should work. See, first of all, they review and finalize the study documents. Review and finalize the study document means protocol. They participate in the protocol, and they prepare the case report form, or we call it pro forma, right? You have heard of this word, like we have to submit the pro forma with the protocol, protocol. So we can, this is a data collection form, this is called data collection form, or called CRF, case report form, right? This is the case report form. So you create this case report form, you create the case report form, you participate in the protocol creation, you participate in the study designs. CDM people start working from the first stage where they review and finalize the study documents like CRF and pro formas, and then based on that they design the database, electronic data capture. They design the data capture forms, electronic data capture forms like in RMS or RevMain or the RedCap. They create the entire design and they give the access to every site or every investigator where they come and they keep those things in the electronic data capture format, right? And based on that, once they do it, they collect entire data, they clean the data, they manage the data, and they track the CRF, like which CRF is incomplete, which site has not filled the full form, I mean full EDC, or which site is giving less data, which site is not able to fulfill that data. So they keep tracking of each CRF until the CRF is not completed by the team. So CRF must be completed by the team. So CDM team takes care of that, and then they enter the data. They enter the data means into the central facilities, like they collect the data from each site and they make a master form, they make a master chart of that form after cleaning and these things. They take the entire data, they make the master chart. In the master set, I mean the EDC only, there is a master chart. They combine all the data and make a master chart, and then again they do the data audition, data audits. Again they do the data audits and quality check. Once the quality check is done, they go for the data validation. Data validation means like suppose site A has given 100%. Out of 20,000, site A has given 100%. So CRAs will go to the site, they will check the physical form, they will check the case files and all, like if the data is correct or not, data is collected correct or not. They will validate the data. They will not check all the 100% maybe, but they'll check 1 2 3 4 10 15%, and they will find out, okay, data is validated, data is correct, not false. Nowadays even every patient is checked. All the patients which are recruited to the clinical trials, they are checked, right? So that is called data validation. Once the data management, data validation is done, they do the discrepancy management. Discrepancy means if there are any problems, data validation. After that, if there are any discrepancies, they manage those things, and they create, I mean, in that final chart, and then they code that data chart. That is medical coding. The here it comes the medical coding. It is also done by the CDM team. They have their own codes, like for the gender, what code they have to give, disease, what code they have to give, sites, what code they have to give. Like this is Australian site is A01, Indian site is I01, Y02, I012, so I12, I15, like that. They give the coding so they can easily subtract the subgroups. They easily define the subgroups and analyze those subgroups, right? And based on that, once everything is complete, they lock the data, and later they extract, analyze, and put it to archivals. So data safety is very important. So data will be safe for 10 to 15 years at least for the archival. If anything happens, if the drug is not performed well or something happens, the governments or regulators go back into the data and check the data for next 10 years to 15 years, right? So that's how the CDM team works. This is called CDM data management process, CDM process. Okay. And CDM process says there are two types of data entry. One is the direct data entry, one is manual data entry. See, so typical approach of data collection in clinical trial is patient, the investigator or the clinical trial assistant, they meet the patient, they enter the data in medical record physically, they transcribe the data on the paper CRF to EDC system. Then the CDM team will clean the data and they will analyze the data. Okay. Another part is they meet the patient, they directly enter the data into metadata, and then they lock the metadata. Data cleaning is done, data analysis is done. So there are two types of format. One is paper format, what we use for the clinical data collection. One is digital format where we collect the data, right? So these are the simple things what you will understand everything here only, or those things are described: data entry, data validation, so managing discrepancies, medical coding, and also data logging. So these are the some tools like EDC, electronic data capture, like the MedRave, Oracle Clinical, RedCap. These are the included data captures where they collect the data, where they create the EDCs, where they create the case report forms, and in that only the data is entered. Then there are some coding standards like MEDRA, Medical Dictionary for Regulatory Activities, or WHO DD, World Health Organization Drug Dictionary for standardizing the data, or maybe the data management systems are there for different data collection and data security. There are some challenges also. So these all everything you will be getting in this CDM course as well. Okay, so AI in machine learning, real-time data analytics, variable technology data, how to retract the variable technology data into the like, because see, nowadays we collect on the paper format, we enter into the EDC, or we directly enter into the EDC. But now the challenges are coming and future trends are coming, like, okay, the person is wearing the variable technology, and the data will directly enter to the EDC without doing any process. So that kind of things are coming in the future for the CDM, right? So yes, guys, that's all from my side for today. And then now definitely someone was asking one class. No, I think this was my last class. This was my last class for this course. And next, I will be taking scientific writing, but next, that after the scheduling, after the ICH guidelines training. Okay. So after that, I'll be taking the class. This was the basic thing what I wanted to tell you, like how to do, what is the cycle, research cycle and all. This is very basic. Okay. So any questions, guys? Any questions? Data validation means comparing the data. Suppose I'm giving the data to you, and you will come back and check the actual source. Actual source means patient files. So that is called data validation. You will match the data. If data is correct, you will lock the data for the final analysis. That is called data validation. So the CRAs goes to the sites. CRAs, what is CRA? Clinical Research Associate, right? So they go to the data sites, they check the data, and then based on that, they find the discrepancies, if there are any misinformation, or fabrication, falsification, plagiarism, if anything, any bogus data is given. So they check everything and do the data validation. Data cleaning. Data cleaning is when the data is filled, they check for missing, like suppose there are some missing variables, there are some data is missing, or some kind of wrong data is entered, right? Or some non-coded data is entered. So they clean the data and make it uniform from everyone. Okay, sir, can you please share the slides? No, dear. No, no slides. Recordings will be there. You will get the recordings. Okay. Yes. Data cleaning, I was telling only, na? Data query and data cleaning, no, those are different. Data cleaning means data cleaning is done before the analysis, so you will get the uniform data. Data query means it can be done from any side. Data query means data query is usually actually done by the CDM team, the clinical data management team. So what they do is, suppose they have some kind of, they find some problem in the data, or they find some kind of, falsification in the data, or some kind of mistakes in the data. So they raise the query to the investigator. Okay, that okay, this data is wrong. Suppose in RMSs, in research management systems, or in these red cap data and all, we have the cap. We have the cap means, like, we have the cap of, like, suppose bilirubin you have to keep. So there is a bilirubin cap, like from 0.6 to like suppose 3.2, 4.2, like that you can fill. If the the cap crosses, then it will show the red. Okay, it will show the red, it will show the warning. And such warnings, the data management team sends the warnings to the team, and then they ask the investigators, they ask, like, why it is, like, in some patients this is exceptionally high, like bilirubin, or the some parameters are exceptionally high. So in those patients, they usually get the queries, and CDM team asks the investigators for the queries. Who performs medical coding? Usually medical coding team is there, or CDM team is there, because there are the MEDRA and there are the dictionaries, so they follow the dictionaries. Medical writers or CDM personnel? CDM personnel, there are medical coders especially. But usually the people who are analyzing, they only code that because they don't make their own codes. There are the codes already for the diseases. Medra is there and other dictionaries are there. Okay. Okay, guys. So take care and we'll see you. I mean, in the classes, someone else will come in the classes, take the classes from Friday. Bye bye. Take care.